Thank you so much for the invitation to talk here at the Humboldt Day today. So my name is Vigdis Vandvik, I come from the University of Bergen, and I'm going to tell you about some work that we have done along altitudinal gradients around the world. So you will see the link to Humboldt right there. My talk has two titles. This is the first. It's related to what we actually study, biodiversity and ecosystems along environmental gradients. And the second title is related to how we did this. We took a plant functional course on a tour around the world. I'll get back to that. But first, let's talk about Humboldt. So this painting from around 1850 was painted by Edward Ender, and Humboldt actually didn't like it because these instruments that are depicted here didn't come out quite perfect. But Humboldt did like to travel. He loved to travel. And back in his time, traveling was really hard, and it took a great deal of effort and a great deal of privilege. Humboldt was super upper class in all the ways that you can imagine, culturally, economically, and so on and so forth. Yet he had to wait for years until his mother died before he could afford to travel and he had the chance. Nowadays, it's much easier. I've traveled to all the continents on the world, or almost anyway, almost all the continents on Earth with science. Yet traveling still does something to us as scientists and as humans. I'll get back to that. But first, this is what Humboldt is known for. Basically, he invited biogeography. He was interested not merely in cataloging plants and animals and places like people were doing back then, but in understanding the processes that determine these patterns that we see, for example, across mountains at different latitudes. He has inspired basically everybody from Darwin to Thoreau to Haeckel with lasting legacies today. And according to Uli Pausas and William Bond, for example, he's to blame for biogeography's infatuation with climate and soil, and its ignorance of fire and grazing as the primary shaping forces in biogeography. And we're still interested in the patterns that Humboldt were describing today. So here's a paper, for example, from this year. And Humboldt was finding patterns, and we're trying to hypothesize about the underlying processes even further, understanding deviations from these patterns. For example, while alpine areas may look very similar in different parts of the world, they still might have quite different functioning. So this paper, for example, is finding that the NDVI, so an indication of the productivity in these systems, are varying quite a lot, both within and between alpine regions. And so we can ask ourselves why. And the work that I'm going to talk about today is research that we have done over the past 5 years to address some of these questions. And this picture is taken from one of our sites in the Svalbard archipelago up in the North Arctic at 78 degrees north. And it's also taken by one of the participants in this work. She calls it the cycle. It's a beautiful picture. And what we'll be talking about now is how these mountains way up in the Arctic are similar or different from mountains at very different latitudes, so much further south. So Humboldt invented isoclines, so these lines that he was drawing across the maps to illustrate how you can have similar kinds of climates in quite different regions, and also how these regions don't necessarily follow strict north to south lines. And while the names that we use for these zones are different now, the basic patterns and the underlying processes that he described, so how temperature and precipitation varies along gradients from north to south and from mountains to lowlands, those are the same. Those are very, very similar. And so we have visited mountains in across a range of regions in the world, from what Humboldt called the equatorial to what he also called the frozen region. Actually, Svalbard, that you saw in the picture in the previous slide, is outside this map. Yeah, and so we have visited quite a number of these regions. We're not showing data from South Africa this time. Those will come in at a later point, we hope. So for each of these mountains that we visited, we set up elevational gradients. And this we did in collaboration with local researchers, so that in each of the sites we studied other factors that they were interested in. For example, this is from Peru, and here we studied fire. We also studied climate change. We studied bird cliff effects in the Arctic. We studied drought. I'm not going to talk about all these details today. I'm just going to talk about the altitudinal gradients. But we set up these altitudinal gradients. We targeted to have around 1,000 meters of altitude. And then we did vegetation plots. So we recorded the species and their abundances in these sites. And we also measured traits of the plants. And so this is what some of these sites will look like. You will recognize the Arctic site up here. Our mountains there are from 0 to 250 meters of altitude. So the mountains are really start at sea level up there. This is from Sichuan in the subtropics, a similar looking landscape, but here we are at 3,000 to 4,000 meters above altitude, above sea level. And then in Peru, we are also at around 3,000 to 4,000 meters. And the temperatures and the precipitation and some of the drivers are different, but the patterns are still overarchingly the same. And so the first thing we can do with these data is, of course, to do some basic descriptions of community structure and how this varies with climate within each region and across them. And so, for example, we can look at things like community evenness that we have here, which tends to increase towards higher elevations in all our regions. And also species richness does the same. And then we have some patterns also along or across the sites where the tropic sites, for example, are more species diverse than the Arctic sites. Now, but as Humboldt, we also want to understand the processes along these gradients and what actually shapes these communities. And so how does community assembly and how does ecosystem processes vary along these gradients? And we used a trait-based approach to do this. So generally, based on theory, and theory going way back, but we have some examples of the more recent expressions of that here. We do expect to find stronger filtering based on environmental variables or environmental factors from the more mesic lowland areas to the more climatically extreme alpine areas. And going concurrent to that, we also expect there to be more intense species interactions, such as competitive interactions, for example, in the lowlands, where there is more resources available. And there's two formulations of these kinds of hypotheses that I have put down on this slide. So one is the stress gradient hypothesis, and the other one is trait driver theory, but there are also other formulations of these kinds of hypotheses. And what's interesting is that these hypotheses makes predictions about what kind of plants we can expect to find at different positions along the gradients. So, for example, at cold or harsh environments or in the alpines, we expect to find species with slow traits like thick leaves, sturdy leaves, long-leaved leaves, and sort of slow nutrient dynamics. Whereas in the more mesic lowland areas, we expect to find resource acquisitive species with traits that make them able to turn over resources quite quickly. So they have thinner, shorter-lived, flimsier leaves than the alpine species. These theories also make predictions about the distribution of traits along these gradients. So the first prediction is, of course, that we expect a shift in the mean values of traits going from one end of the gradient to the other one, based on these general relationships. Another prediction is that we expect differences in the variance, for example, where we expect species filtering at high altitudes to result in quite narrow range distributions of traits based on environmental filtering, whereas we expect in the lower on the gradient, where we have more competitive interactions and limiting similarity, then we expect a more broadly, even over-dispersed distribution of traits. So these are, of course, hypotheses that we can test using the data that we have collected. And so across our regions, or the 5 biomes that we have been studying, we find some trends both within and across these biomes in terms of traits. So in these plots, I'm going to show you some of these plots, and here I start out having mean annual temperature as the x-axis, and then different mean trait values as the y-axis. And one of the things that we see here is that there seems to be, and here are our 5 regions, there seems to be some patterns in mean traits. So for example, plant height, the upper right-hand plot there, there are much shorter statured plants in the Arctic than we have in the tropics, and the same thing with altitude or with temperature going from the warmer lowlands to the colder alpine areas in each of the sites. There we also have the similar kind of trend. Now, one of our regions stand out, and that is Colorado, which tends to be doing the opposite thing a lot of times in these graphs. And Colorado happens to be in a quite dry region, and so therefore these sites are, or these gradients are drought limited rather than temperature limited, and so in a way things are turned around. So already we see that alpines are not necessarily the same, or mountains are not necessarily the same in these different regions. But except from this Colorado exception, we see that the patterns along the mountain gradients match up rather nicely, especially for size-related and growth-related traits like height and leaf area and leaf thickness and dry mass. All these regions line up quite nicely. When it comes to traits like specific leaf area and leaf dry matter content, so traits that are related to this fast-slow hypothesis, or this fast-slow dynamics of leaves, there seems to be more inconsistencies, however. So we see that some of these local gradients, for example in Norway and in China, are actually going the other way around compared to the overall trend. Now, we put mean annual temperature on the x-axis here to start with, and this of course conflates 2 things. Firstly, it gets colder as you go up a mountain, and it gets colder as you go up north, but there's also a lot of variation in seasonality between these sites. And so, for example, in the Arctic we have a growing season of 2 months, whereas in the tropics we have a growing season that lasts the whole year. So another way of lining these mountains up or comparing them is to look at the temperature that the plants actually are exposed to when they grow. So if we change our y-axis or our x-axis to be the mean temperature of the warmest quarter, so basically parts of the growing season, then the patterns look a little bit different. Again, we see that the size-related traits seems to make sense. But what we see quite clearly now is that when it comes to these fast-slow dynamics traits, it seems like the tropical sites generally have more stress or slower dynamics, or sort of slower turnover traits, or more stress adapted traits for the same growing season temperature compared to the higher latitude sites. And one possibility of this is, of course, that it's related to the conditions during the growing season. So maybe it's related to more mesic growing seasons in the short northern summers. It can also be related to seasonality in drought in the southern climates. So these are things that we are interested in exploring more closely. We also talked about predictions when it comes to variance. So we said there would be higher variance in mesic areas where you have more species interactions, and lower variance in more extreme area climates where you have more environmental sorting. And this is also something that we can explore in our data. So again, we have mean temperature of the warmest quarter on the x-axis, and what we see is that for the vast majority of these traits, we have lower variance both in the north, so in the more extreme climate in the north, and also in the colder parts of each of the gradients. So this seems to match up quite consistently, suggesting that the relative importance of plant-plant interactions and niche partitioning is increasing towards warmer climate, both on the geographic scale and also down our mountains. Another question that relates to Humboldt's hypothesis and to his explorations of mountains is, of course, asking to what extent are these alpine systems that we study just functional extensions of those systems that we find at lower altitudes. So are mountains basically just a continuation, or the alpine grasslands basically just a continuation of the forest down there? And in Peru, in Huayquecha, we were very lucky to have the possibility to explore this. Because the ABERG, the Andes Biodiversity and Ecosystem Research Group, have been working along these gradients for an extended amount of time. And what they have been doing is that they have been setting up plots from way down in the Amazon at very low elevations and all the way up to 3,500 or over 3,000 meters of altitude in the Andes. And we combined these data that they had with our new data on the Puna grassland gradients. And so here is we just put this together, so these are some quite new data, and I'm not going to go very much into details about interpreting them, but I do want you to look at the x-axis of these plots, because here we have 4,000 traits gradients that are 4,000 meters long. That's quite interesting, and all these are traits that are measured on site in our study systems. And one of the things that we see here is that when it comes again to the fast-slow sort of continuum traits, there seems to be some quite marked breaking points between the forest plots, which are in green, and the Puna grassland plots, which are in purple. So in the forest, when you work your way towards the alpine, you go from more resource acquisitive towards more resource conservative traits, as you might expect. But there is both sort of a step difference and also a slope difference when you come to the Puna grassland. So something seems to be happening there. Interestingly, when it comes to the nutrient traits, so for example, nitrogen to phosphor ratio, which is indicative of nutrient dynamics of the system, things seems to be very consistent. So this is something that we will be exploring more in the past or in the future. Stepping back a little bit, I've given you a taste of some of the first analysis that we have done of these plant functional traits along these elevation gradients across the biomes. And these are data that we are in the process of exploring in much more detail, both in relation to these gradients and also to these experiments on fire and climate and bird cliffs and so on that I was talking about earlier. And to give you an idea about the kind of data that we have, some quick statistics. We have measured the specific leaf area of more than 18,000 leaves. We have surveyed more than 5,000 vegetation quadrats. We have scanned and measured more than 15 square meters of leaves. And we have collected 1.2 kilos of dry mass of vegetation that we have taken back for chemical analysis and so on. And if you're not too impressed by the last number, remember that we are working in the mountains here, and so some of the things that we are working with are quite small and quite hard to work with. So how have we done this? Well, our secret is to be many. And so if you saw on my first slide, it said some names, some people that have actually contributed plots or analysis to this talk, but it also said that there were 105 other people that were involved in the work. And so these are some of these people. And what we do is that we travel around to different parts of the world and measure traits, but we also measure ecosystem and physiological responses that I'm not talking about today. So these are some of the people in Svalbard and Peru. Here are some more people, also in these two same localities. Collecting these quite large amounts of data with a lot of people involved have taught us to be really meticulous about our protocols, our data documentation and training. Reproducibility is of course a hallmark of science, and being reproducible when you work with these amounts of people and traveling around the world with different groups of people is very difficult. So this is also something that we are really focusing on with all the participants on all these campaigns. We also like to do our rainbow pictures, by the way. So that's also something that we repeat across latitudes, but I won't talk about that today. And the way that we do this is that we do it as part of a plant functional traits course. And this is from our web page showing, first of all, the regions that we have had courses in, and then also the number of participants that have participated from different regions. And you will see that there tends to be sort of a hotspot of participants from where we do the courses, and that's of course because we involve people from those regions in the courses. So we prioritize both students and teachers from those regions in the different courses. We've been doing this since 2015. And so our focus in these courses is to teach and learn the theory and background of various aspects of trait-based ecology, and the participants then plan and conduct and report on the results from these courses. And then together with everybody who has been to these courses involved, we publish the data coming out of the courses, and then we also encourage the students to publish some of their group projects from these courses. And so this paper, for example, was led up by John Henn, who was one of the students on the China course. And I'm not suggesting that what we do here compares to Humboldt's masterpiece Cosmos in any way or form. But there is one parallel here, because he was also surrounded by an army of students and helpers who collected data and read and wrote for him. But they didn't get to be on the front page of the book, or indeed anywhere mentioned in the book for the most part. And so what we have gotten better at in science over the past 250 years is by giving and sharing credit. And this is something that's really important, and this is something that I think we should think very carefully about when celebrating people like Humboldt, which we also should do, but we should also recognize that science is a living anthill. It's not the memorial sculpture park. So we need to recognize the contributions of the participants in science, all of them. And we also need to recognize that we are building standing on shoulders, but building forward together. And learning and doing and collaborating over science can be a life-changing experiment, surely, but also experience. And our students, so these are some of the students some of the statements that our students have said after participating in the course. And what they show is that doing science collaboratively like this can be a life-changing experience way beyond science itself. And so, for example, doing good research just means having yourself together as a person, which can be really hard too. So it's sort of it goes way beyond science. And I just wanted to show you this because these are Dumble plots, and basically what they show is what the students were expecting coming into the class, or into the courses. So that's the left-hand side of the point, and then what they were reporting going out. And what is interesting is that our students, they are expecting to learn useful field and lab techniques doing these courses. But the collaboration and being in an international setting with other researchers and also working at the field station is something that really supersedes their expectations. So they were positive at these things before, but they're really sort of much more, they have gained much more than they traveling with science, as Humboldt was clearly experiencing, and that was really, really important for him and his way shaping the way that he was thinking about nature and life and the universe. Certainly also has a power today. And so with that, I would like to thank everybody involved, obviously, and our funders and our home institutions and our collaborators, especially in China, Peru, and Svalbard. And then everybody who's been involved in addition to the people who participated in the courses. And my 111 co-authors on this talk, which will be here in a second. And then, of course, also all the hard work that went into the data collections. So here is everybody. With that, I would like to thank you so much for inviting me to give a talk on the Humboldt Day, the 1 week, the whole week Humboldt Day, and I'll take any questions. Thank you.